The overall goal of this procedure is to follow activation of RhoA-specific GTP Exchange Factors, or GEFs, in cultured cells by making use of a mutant RhoA GST fusion protein that has high affinity for activated GEFs. This is accomplished by first producing the GST-tagged mutant Rho protein in bacteria. Next, the bead-coupled GST-RhoA is purified. Then, the purity and concentration of the GST-RhoA protein is assessed using SDS Page. Finally, cell lysates are prepared and a GEF precipitation assay is performed with the GST-RhoA protein. Ultimately, results can be obtained that show increased precipitation of activated GEFs in treated cells through Western blot analysis of the precipitated proteins with a specific anti-GEF antibody and quantification by densitometry.
This method can help answer a number of exciting questions in the Rho GTPase field, such as how are the many Rho GTP Exchange Factors regulated in a signal-specific manner, and how is their activity coordinated.
Generally, individuals new to this method will struggle initially because the GST-Rho G17A protein is relatively unstable and can break down during preparation. It is also important to remember to perform the GEF precipitation assay quickly and consistently.
Demonstrating this procedure will be Faiza Waheed, a master student from my lab.
To transform E. coli, quickly thaw an aliquot of DH5-alpha competent cells in an ice bath. Add 1 microliter of 25 to 50 nanograms per microliter.